let's continue with our discussion of metabolism. This is Metabolism Mini Lecture B. Gluconeogenesis. What in the world does that mean? Gluconeogenesis. That means making glucose anew. Neo means new, and genesis means to make. So gluconeogenesis is the synthesis or production of glucose from pyruvate, lactate, glycerol, alanine, or glutamate. So remember, alanine and glutamate, those are amino acids. We talked about pyruvate and where we get that, right? That's from glycolysis. So that could be the result of anaerobic respiration. And lactate or lactic acid is also produced in anaerobic respiration. You do not need to memorize all these steps. Just know that we can make glucose again from different molecules, different metabolites, and for the most part this is going to happen in the liver. All right, we can also use triglycerides, so we should remember triglyceride molecules, right? We've got 3 fatty acids. In this case, we've got different chain lengths on those triglycerides, on those fatty acids, and then this is our glycerol head. So a triglyceride is for long-term energy storage. So we're not going to get into this until we've used up our glucose and used up our glycogen, right? So this would be stored in our adipose cells, right? And we'd get it in our diet as well. But when it comes in from the digestive system, it's going to be coming in as fatty acids and not full triglycerides. All right. Chylomicrons contain these triglycerides. They're cholesterol molecules and other protein molecules, apolipoprotein, so that's going to be something with both a lipid and a protein portion. And they're going to carry these water-insoluble molecules from the intestine through the lymphatic system and into the bloodstream, and then carry those lipids to the adipose tissue for storage. Because remember, our lipids are not water soluble. So if we're, let's say we had our mac and cheese with plenty of triglycerides in there, or maybe some fried food in our digestive system, we can't just take that fatty acid out into the bloodstream. We're going to carry it in this this little gadget, this chylomicron, right? All right, breakdown of fatty acids. During fatty acid oxidation, triglycerides can be broken down into acetyl-CoA molecules and used for energy when glucose levels are low. So remember that acetyl-CoA, we're going to use that inside the mitochondria. So we can break down our fatty acids when the glucose levels are low, right? So that's when we haven't eaten. Maybe there's been a long-term famine. Maybe someone's dieting, maybe we're exercising a whole lot, but when our glucose levels are low, we can get into those fatty acids and use them in cellular respiration. All right. Excess acetyl-CoA is diverted from the Krebs cycle to the ketogenesis pathway. Right. So remember our stages within the mitochondria. First we made some acetyl-CoA, then we had Krebs cycle, then we had the electron transport chain. So excess acetyl-CoA can be diverted to the ketogenesis pathway. Occurs in the mitochondria of liver cells, right? Liver cells, right? Liver is doing a lot of things for us. The result is the production of beta-hydroxybutyrate, primary ketone body found in the blood. So when we don't have enough glucose, we can use that ketone body, can be oxidized to produce our acetyl-CoA, right? So then we can use that in the Krebs cycle, right? So we can use that in the Krebs cycle. All right, lipid metabolism. So we can do different things in lipid metabolism. Glycerol and fatty acids follow different pathways. I'm not too worried about this at this point. Right? We do want to know this: digestive enzymes and hormones. Digestive enzymes and hormones. Enzymes in the stomach and small intestine break down proteins into amino acid. Hydrochloric acid in the stomach aids in proteolysis, and hormones secreted by the intestine cells direct the digestive process. So we'll talk more about digestion as we go along, but in the mouth we have amylase, which is a starch, is an enzyme that's going to break down starch, and that's going to help that's why we like our carbohydrates, right? If we're eating starch, it's going to taste sweet as we're chewing on it. In the stomach, we have proteases and we have hydrochloric acid. So we're beginning carbohydrate digestion in the mouth. We're beginning protein digestion in the stomach with the proteases and the hydrochloric acid. Now, just as we leave the stomach, we're going to have the pancreas, right? So the pancreas is right behind the stomach, and this little duct is right here. All right, let's see if we can point that out. Is right as the food is coming into the small intestine. And here we're going to add several different enzymes, right, that are going to break down protein, break down lipids, break down carbohydrates. We're also going to add our sodium bicarbonate, which is going to neutralize that stomach acid. We're also putting in our bile that's going to help emulsify the fats. So at this point, we're dumping in all of this stuff into the small intestine to help break down that food into the smaller particles or smaller molecules. So remember, we have our macromolecules, our proteins are broken down into amino acids, our carbohydrates are broken down into sugars, our lipids are broken down into fatty acids, and our DNA, or if we're eating cells, there's going to be DNA and RNA in it. There's going to be nucleic acids in it. Those will be broken down into nucleotides. All right, that was mini lecture B.